For this video, we will cover how to complete one-trait Punnett squares, specifically those that relate to X-linked traits. One of the biggest differences between X-linked Punnett squares and autosomal Punnett squares is that we have to keep in mind the sexes of the individuals in our cross, and we have to use X's and Y's to indicate their genotypes. Also, we are only going to be worried about X-linked recessive traits for this class. So let's quickly run through the 5 possible genotypes. We have big X, which would be a full dominant female. These would not have the trait. We have a big X, which would be a heterozygous female, and note that these are also considered carriers. Carriers are individuals that carry a trait but do not express it. Then we have x, which is a recessive female. These would display the trait. In terms of the males, we have Y, big X, which would be a dominant male that does not express the trait. And we have Y, x, which would be a recessive male that expresses the trait. Note that since males only have one X chromosome, they either express the trait or they don't. And as such, we cannot have carrier males. Now let's get into actually solving these Punnett squares, and let's use colorblindness as an example. Let's have a cross between a normal vision male, Y, big X, and a carrier female, big X. We set up our square, and let's add in the potential alleles each member can give off. Now let's multiply each set of alleles against each other to solve the Punnett square. Top left is Y, big X. Top right is Y, x. Bottom left is big X, and bottom right is big X. At this point, we have technically completed the Punnett square, but there are normally a set of questions associated with them. The most common question is simply, what is the percent chance that a certain genotype or phenotype is seen? So let's go ahead and calculate all of them. Let's make a quick list. We could have colorblind females, carrier females, and normal vision females. We could also have colorblind males, carrier males, and normal vision males. As mentioned above, a carrier male is not possible. However, they're often included in the calculations because teachers are fun like that. Luckily, we can just go ahead and make it a 0% right away. Now let's go back and look at the others. Looking at the pedigree, we can say that we have one normal vision female, one carrier female, and no colorblind females. And since there are none of these, we can just go ahead and say that is 0%. We have one colorblind male, and we have one normal vision male. And once we get to this part, the math can get a little funny. Often questions are specific, asking, for example, what is the chance of having a colorblind son? Now, we have specified sons, and as such, we can only look at the sons in the square. So let's go ahead and white out the females real fast. Now we only have 2 boxes, and because of that, the colorblind individual represents 50%, and the normal vision individual represents another 50%. We can then flip the question and worry about the female individuals. Here we would white out the males, and we would end up with 50% of the females having normal vision and 50% being carriers. Let's go ahead and reset real fast and try a different cross to practice some more. We're still going to be using colorblindness though. So now we have a colorblind male, Y, x, and a carrier female, big X. We set up the square, we add in the potential alleles each can give, we multiply our alleles against each other, and we get top left as Y, big X, top right is Y, x, bottom left is big X, and bottom right is x. Let's make our list. So we have colorblind females, carrier females, normal vision females, colorblind males, carrier males, and normal vision males. Now let's look back at the square to see what we get for the males. Again, the carrier will be 0%, and we again get 50% colorblind males and 50% normal vision males. Now let's look at the females. Here we would have 50% colorblind females and 50% carrier females, which gives us 0% normal vision females. As you go through and complete these types of Punnett squares, make sure that you follow the basic rules for setting them up. Set up the square and add in the alleles from each, multiply the alleles against each other, then do the math for both the male and female individuals. This concludes our video covering how to complete one-trait Punnett squares, specifically those that relate to X-linked traits.